Sir, sir, how many hanger? Download பண்ணி
இல்ல சார் அது லிங்க் ரெக்கார்டிங்காக it's already folder we have put it everybody's
one slide na
2 minutes.
2 minutes da okay madam
Respected Mr. Good afternoon, all of you. And my title is Food Plot Management in Mulberry using the Actinobacteria bioformulations. And root rot, you are all of you that root rot is the major disease which causes around loss of 70%. One it is being affected, the entire crop get affected because disease. In Tamil Nadu, it is a serious one. Hence, we conducted one survey, and survey in the area of only Erode, Coimbatore, and also Pilpur district. In that, we have conducted survey and also recorded the incidents, and we collected samples from all the area. We have isolated with three types of pathogen. Last we program actually several types of food are there, but in case of the Tamil Nadu, we recorded 3 pathogens. So last we program macrophominate also is the rhizotonia, and in this among that among the surveyed area and the highest incidence of 31% incidence, it was recorded in the Kundadam area of Tirupur district. Hence, we collected the samples from that particular area and isolated, and the pathogen is Lactobacillus. And for this metals method, first I started isolation. Second one is the screening of actinobacteria and morphological identification and preparation of bioformations. In that, after the culturing the Lactobacillus, we have characterized the morphologically and also molecularly. Then later, actinobacteria isolates we have collected from the forest region, rhizosphere region of the forest area, from the forest college and Nitpalayam, from different trees, tree, neem, eucalyptus, and the tashrina like that. From different rhizosphere region, we have collected 7 isolates, and we have screened against by dual plate technique. Among that, all isolates, 7 isolates we have isolated. Among that, A7 isolate it was recorded proved to be highly efficient based on the dual plate method. After that, we extracted from the root extractor. It was utilized for against for the screening by using the agar-well diffusion technique, and also this agar-well diffusion technique at different concentration to extract the molecular activities. And also we identify the effective pathogen by the molecular identification. By the molecular identification, it was proved by the that effective isolate was identified as the Nocardia morphologensis, and this is the ID number, and we have submitted in the NCBA database. We have submitted the culture and this one, and after that we have prepared the formulations based on there are 3 different formulations: salt formulation, seed formulation, and pupil powder formulations. And all the formulations that were prepared and also tested, and at a different rate. 5 gram, normally 5 gram is better, and also for to test the efficacy, both 5 and 10 grams. First one is salt formulation, then the peat soil and pupil powder formulations. When it was actually the test was carried out in conditions, classes and also in condition. But the data space I put only the greenhouse, and also in the greenhouse before besides we recording the disease incidence, it was also the growth parameters, yield parameters also recorded. Among this, the seed formulations at the rate of 10 gram when we are applying to the seedlings, it proved to be the disease incidence was recorded the lower incidence of 8.62 compared to control it is 81. It was recorded 1 month after the applications. And also the yield parameter, yield and also the stem length and also root length, it was recorded under the glasses condition. These things, the feed formulation is proved to be maximum, and it may be the reason because of what is the feed also contains some organic nutrient material which may support the plant growth beside this one. And also when we have started the shelf life period in of the different formulations. Among the shelf life formulation was from 30, 60, 90 and also 120 days. And the pupil for the feed leaf, it maintain the reasonable populations of the 2.69 into 10.5 populations on the 19th day. Normally, bioformulations are for Trichoderma everything. We can store the period only up to 3 months. After that, it is slowly degraded. Hence, we compare this one, it's up to 90 days. It is very good support we are receiving in the feed formulations. Hence, and also we have connected the field trial also. Field trial we recorded the result. In that also it recorded it gives a good result using the feed formulations. Hence, once the conclusion is we are applying the feed soil formulations using that effective isolate, it will be highly effective in the management of the root diseases. This is my stand.
Which is what which type of root practical? There are two types of
Different type of root roots, but in the surveyed area we have collected that the glass is not.
black,
Black root.
black root. Because white to violet is there.
White.
Is it not because of melodina? This is called melodina.
Not chemicals, sir. This is. Actually, as per the code of the actually formulates were told, no inani, no melody, and try to cover all these things. That's all. Thank you.
Next question. Next, I will invite other consideration India. And next week, teaching additional
Thank you.
present genetic diversity studies based on principles of analysis for morphological traits in mulberry genotypes. Yes, ma'am.
The topic was genetic diversity studies based on principal component analysis for morphological traits in mulberry genotypes. We are already known mulberry is a sole crop of silkworm as well as the multi-purpose utility. If we improve the social economy of farmers, we develop a new varieties. For this view, we have to go for identify the diversity of the parents. If parents having more diversity means more contribution of characters towards the one generation to another generation was more. So we have to read the new varieties as well as the hybrids. So PC analysis generally used to grouping the data in mulberry genotypes, which will be enable breeders to classify the available germplasm into distinct groups based on the genetic diversity. In methodology, in Department of Sericulture, Metropolitan having 100 number of germplasm we have established on the maintained. That was obtained from CSERC also as well as CSRPI also. From that one, I have taken 22 number of mulberry germplasm from 5 different species: Morus indica, alba, Lavilletta, latifolia, and the Catayana. It was raised in 3 replications. I have taken some of the traits like qualitative traits that was governed by qualitative traits such as plant figure, plant growth type, shoot type, mature fruit color, leaf angle, leaf shape, leaf color, leaf angle, and a leaf phase. From that one, principal component analysis, there are 4 components contribute the 70% of variability. That means more variable means that will heritability also more in that case. So the 4 component in that first component, the plant figure, shoot type, mature shoot color, leaf angle, and leaf shape was contribute more variable. So from that holistic, based on the mean of first 4 PC component, plant figure, shoot type, leaf angle, leaf shape, and leaf hardness are most important traits contribute in the overall variability. If characters with high variability are expected to provide high level of gene transfer during breeding programs, this is an important backup for breeders to investigate trait specific varieties or hybrids in mulberry breeding. Thank you.
Next, I invite enhancing silk productivity in Bobby's Murray through nanoparticle treatments, optimizing yield and quality parameters.
Good afternoon to all. Today I am going to present my part of famous research work, which is entitled on enhancing silk productivity in Bobby's Murray through nanoparticles treatment, optimizing yield and quality parameters. Let's move on introduction. Sericulture is a remarkable agro-based industry. Nowadays, the farmers are facing a lot of challenges to produce a good quality of food because of poor quality production of leaf, sudden outbreak of pest and disease incidents. In that, many researchers are trying to resolve it. In that, nanotechnology is one of the innovative technology to resolve it. In my research, I have taken the two components. One is spirulina and another one is titanium dioxide. Spirulina, generally, you know, spirulina is a single cell protein which contains 18 amino acids. This amino acid is used to produce good quality of food and pivotal vitamins also. And titanium dioxide is used as a nanocatalyst. And it is used to feed additive and protein synthesis. Let's move on methodology. First, I had taken the 80 ml of aqueous solution of spirulina and mixed it with the 20 ml of 0.01 millimolar of titanium dioxide. With the help of hot-plate magnetic stirrer up to 6 hours, this solution is turned to pale yellow color. Through visual identification, the nanoparticle has formed. In scientifically, we have to characterize. Then only we have to confirm the nanoparticles. In this research, I analyzed 6 instruments such as UV visible spectroscopy. This spectroscopy to identify the nanoparticle with the help of wavelength. In this nanoformulation, it absorbs 230 nanometer. Then followed by particle size analyzer. This particle size analyzer is used to identify the particle distribution and size. In that, with the help of zeta potential minus 20.08 millivolt, it's clearly said that the particle shows normal distribution and the size is 97 nanometer. Then followed by FTR. This FTR used to analyze the functional group of the nanoparticle. In this formulation, we identified 30 functional groups. In that, amine groups are enhancedly available. And followed by XRD. This XRD is used to identify the crystallinity of the nanoparticle, which has recorded the peak value of 27.72 degrees. Then followed by SEM and TEM. This SEM and TEM used to analyze the shape and the size of the nanoparticles. It's clearly showed irregularly dispersed and hexagonal structures as clearly observed. Then followed by EDAX. These EDAX identify the nanoparticle, which are the elements present in that nanoparticle. I have showed this titanium dioxide is high, then followed by sodium, magnesium, zinc, and minor nutrient also available in the nanoformulation. After confirming these nanoparticle characterization, then go for bioassay. This bioassay, I tried the different concentration of the nanoparticles from 25 to 150 spirulina alone, titanium dioxide alone, distilled water, and control. In that, 50 ppm shows a best value compared to all treatment. Because of higher concentration of titanium dioxide, it leads to the toxicity to the silicon. That's why 50 ppm is best compared to the other treatment. My conclusion is 50 ppm gives more yield compared to the other treatment, which increases compared to the control 10% is increase. Thank you.
Questions?
Post also oral presentation.
Yes, it was.
Okay. Sir, think like, this is your presentation. Your only. Earlier you said when is carried in said.
Okay, next. I call Mr. Professor Alban to present on the impact of zinc and ion spraying on growth and yield of mulberry vegetables.
Slideshow.
Sir, the thing is, the poster is regarding preparing a product for increasing the yield and quality of both mulberry and silk. This is the main aim of the study. And what we have done is, that micronutrients is playing a critical role in increasing the yield and quality of all the forms of life, you can say. That's the reason why I have chosen this topic and I am presenting this one in this forum in the concept of One Health perspective. Because the soil has to have enough micronutrients, then only the plant can take up. And if we feed this plant produces how this animal and human nutrition is improved. This is what, so with this aim, we have taken this study to optimize the concentration of zinc and iron, which are more important for improving the yield and quality of silkworm and mulberry. So as all sericulturists, I am a soil scientist. I'm telling the mulberry plants are rich in phenol, oxalate, and phytic acid. These are all the three anti-nutritional factors which reduces the assimilation and also utilization of these assimilated micronutrients in the leaf. That is the main reason for reducing the quality of the mulberry leaf, and it is a key and quality barrier present in the mulberry leaves, which is the main source for improving the productivity and quality of the cocoons in mulberry silkworm or any other silkworm. So what we did is we want to break the barriers of these anti-nutritional factors by imposing this zinc and iron through fortification of mulberry leaf. And another one is by feeding this fortified mulberry leaves, how it is imparting or influencing the yield and quality of the cocoon. This is what the purpose, for this purpose, we have carried out 2 season trials. And this is the, I don't know, please. This is the pooled values of both the experiments. And we have taken a concentration of 2 concentration in zinc, 0.25 and 0.5%, because optimally, in many of the crop, 0.5% zinc sulfate is the optimum dose for increasing the yield and quality. Whereas in the case of iron, in agricultural crops, they are going up to 1%. But in mulberry, they are saying that 0.5% itself is enough. But the present study, what we have done it individually, we have tested this zinc sprays at different concentration, iron spray in different concentration, and combination of both iron and zinc we tried. And we found that individually, zinc is having the better results than iron in increasing the quality and the yield of mulberry and silkworm. This is the finding we got it. And with regard to iron, the higher concentration of iron is having the negative impact on the yield and quality of silkworm and mulberry. So particularly at 1% concentration. So the best treatment what we have got from the study is 0.25% of both zinc sulfate and iron sulfate combined spray, or 0.5% zinc sulfate or 0.25% iron sulfate. This is the finding what we got it for improving both yield and quality of mulberry. And after fortifying this leaf, we fed this, please go ahead. We got around 23% of yield increment over normal practice by going with this zinc and iron spray. And we have also conducted a biosis study with the Krishna the double hybrid. And we found that the enhanced larval weight and cocoon weight was observed when we are feeding the leaf sprayed with 0.25% of zinc sulfate and iron sulfate. The difference, normally people are working, people are working individually, directly spraying the leaf with different concentration of micronutrients and feeding that leaves to the silkworm. But when we are going with this type of enrichment in the natural way of cultivating the crop by adding different concentration, we can get better utilization, better assimilation, and also improved version of all the yield and quality in the silkworm also. And the feeding behavior is also differing when we are going with spraying directly the leaf and also the enriched leaves. This is the difference what we observe. And we also found, I haven't produced all the data what I generated out of this study, but what we have found is the yield barriers, the anti-nutritional factors are reducing the enzymes, particularly with zinc and iron, the peroxidase and superoxidase mutates. These 2 enzymes are the most important enzymes, increasing the zinc assimilation and utilization in the mulberry leaf and fortification. This is what we observed. And in the case of silkworm, we found that the incidence of diseases and pests. This is also reduced lesser when compared to the controlled leaf what we fed to the silkworm. This is what the outcome we have generated. We want to develop it as a product, as a holistic product. So we just find out the concentration which is optimal for increasing both yield and quality of mulberry and silkworm. And maybe in the future experiments, we will be collecting all those things, and we will be adding some more anti-nutritional breakers, like to reduce the phytic acid, zinc is having the significance. Whereas for protein synthesis also, zinc is having the impact. So that's the reason why we have taken this one, and we need to go with some more vitamins and other things also in context of silkworm to improve the quality and productivity of the silkworm and mulberry. Thank you. Any questions?
Any questions?
No more questions. Thanks so much.
Next, Okay, the Priyadashini can present on mulberry leaves and silkworm excreta as a source of natural colorants for dyeing of fabrics.
Mulberry leaves and silkworm excreta as a source of natural colorants for dyeing of fabrics. Actually, we know that after feeding of silkworm, mulberry leaves are left in the mulberry garden, as well as silkworm excreta after completion of silkworm rallying disposed as a waste. So created environmental nuisance. So though it is rich in natural coloring agents like beta-carotene, lutein, and quercetin, proper utilization and recycling of these natural resources have become the need of the world. With this, so we have extracted the dye from the mulberry leaves as well as silkworm excreta. For this, 50 grams of mulberry leaves taken and boiled with sodium hydroxide as well as water, and the solution was allowed to cool and filtered and used for dyeing fabrics. Here we have used different types of fabrics like cotton, silk, and wool that was treated with pre-modernity, meta-modernity, and post-modernity for 40 minutes at temperature of 85 degrees Celsius and dried. After this, the color strength of the dried fabrics was computerized. Coming to the results, actually the dye extract yield from mulberry leaves was 16.95%, and from silkworm excreta it was 17.56%. After this treating with the fabrics, the highest color strength was obtained by pre-modernity method with woolen fabric in copper sulfate as a model. Both in mulberry leaves as well as silkworm excreta, the color's high strength value is 184.91 and 82.05. We can see the different shades like in copper sulfate based model like tallow, thick, tobacco, brown, pest, lime, oak, and clay brick. So from these studies, it is concluded that instead of going as a waste, we can use mulberry leaves as well as silkworm excreta for extraction of dyes, and instead of using of synthetic dyes, and it also it is rich in colorants. It can be used in pharmaceuticals as well as food industries as a natural colorant.
It is assessing the impact that registered seed producers on sericulture development in Australia. So,
WhatsApp.
Assessing the impact of registered seed producers on sericulture development in West Bengal.
our government is mainly focusing on employment generation and the private participation. In that, from last 10 years, Central School is developing a research team for commercial seed production. One point, then from 9 that 1,000 metric ton production to now currently 2,000 metric ton has been increased. With these uses of traditional sericulture combined with the old people, they are going with the sericulture. In that methodology, we conduct a survey only in the region of Maga, major sericulture. So we selected the Malda region. Among the lead the data production of different fibers, that is Savani, Bagmi, and Agrani, Bagmi. Every year to contribute to the silk production. And this private participation is nearly 70% they are contributing, remaining 20% from the central silk board and 10% from the state unions. So in that study, we are concluding that private participation and private RSPs are coming up for taking the seed production because it is giving them more income from the seed production and quick supply to the farmers.
ವೆಸ್ಟ್ ಆ ಕಡೆ ಬೆಂಗಾಲ್ ಈ ಕಡೆ ಬರ್ಬಾರ್ದು ಸೌತ್ ವೆಸ್ಟ್ ಬೆಂಗಾಲ್ ಒಟ್ಟಿಗೆ ಇರ್ಬೇಕು ವಿಜಯ್ ಹೌದು ನಮ್ಗೆ ಗೊತ್ತಿರೋದು ಹೇಳ್ತಾ
Any questions?
ಎನಿ ಕ್ವಶನ್ಸ್
Oh, there maximum private.
Maximum.
ಪ್ರೈವೇಟ್
More or less 1 crore there.
ನೀವು ಎಲ್ಲಿ ವರ್ಕ್ ಮಾಡೋದು ವಿಜಯ್ ವೆಸ್ಟ್ ಬೆಂಗಾಲ್ ಅಲ್ಲ ಕನ್ನಡ ಗೊತ್ತಾ ಮದರ್ ಟಂಗ್ ತಮಿಳು ಇಲ್ಲೇ ಅಲ್ಲಿ ಹೋಗಿ ವರ್ಕ್ ಮಾಡ್ತಾ ಇದ್ದೀರಾ ரொம்ப நன்றி யூ கேம் ஆல் தி வே ஃப்ரம் வெஸ்ட் பெங்கால்
Next slide.
விஜய் இந்த தமிழ் விஜய் ஃபேமஸ்
Dr. Shanmugam, Professor, to present on introduction and evaluation of tropical Tassar silk farm varying on Arjuna, the western Ghats of India.
ವಿಜಯ್ ಕನ್ನಡ ಹೊಸ ಪಾರ್ಟಿ ಕಟ್ಟಿದ್ರು ಅಂತ ಇದೇ ತಮಿಳ್ನಾಡು
My topic is an introduction and evaluation of tropical Tassar silk worm, varying on opportunities in the western Ghats of Tamil Nadu. Yes, I involved the work is to find out the suitable Tassar silk worm for Tamil Nadu condition. For the experiment, I have taken Tava silk worm base, which is purchased from the BTSS fellow store. And for the experiment, I have taken a plant that General Arjuna. The experiment conducted at the Department of Sericulture, Forest Institute, Tamil Nadu. And then coming to the key results, the development of duration of embryo myelita, such as period 10 days, then larval period of 23 days, then followed by puberty period of 23 days, and adult period is 8 days. Here the main thing is particular rate of rally, that is reached up to 40%. Then coming to the cocoon characters, the cocoon weight reaches up to 9 grams, then followed by pupil weight is 7 grams, and shell ratio, I mean the shell plus shell content is that is 2 grams, then followed by shell ratio is 22.25%. That's all about my. Thank you.
Any questions? Sir, we can also.
just
Yes, madam.
sir
Yes, sir, please.
serial number
FCRL, sir, 11.
11 Arjuna, tomato soil is not working effectively.
This is a preliminary study which you can study with the thermal Arjuna.
Arjuna, we have Arjuna.
Next, I invite Tangavala.
Balarika.
Present an impact of alternate participants on difference and performance areas.
Field work is sir. Field
Good afternoon to everyone.
Good afternoon.
Madam, payroll.
Angamala.
No, like serial number
8 months.
8.
Actually, it's the corona period, just to some interest, actually continuously, we can get a silkworm in a metropolitan region. But the availability, source of castle is very scarcity is more. So those type, so I just whatever tree crops available inside of forest college. So I just tried to rare the yellow silkworm by 3 host plants. So actually it is a multi-voltane, another one thing it is a polyphagous. So though having already more than 15 host alternate host plants are there for any silkworm ready. So my thing is, so alternate, what is the suitable alternate host according to for alternate for cluster. So just I tried several crops. So tapioca, then jatropha, then terminal arjuna, terminal katapa, and sampaka. So then Dixa. So these crops are available. Exception tapioca only I just collected from outside. So those things I what are the crops is preferred by the most preferred by the area silkworm. So almost cattle 100% regular preferable. Then next is the tapioca. Then jatropha almost 70%. So other minimum is actually Pixa, cassia, and some other glycidia. So those crops is not preferable by the silkworms. So almost even after I tried, so first is up to final instar, but first 2 instars it is almost it will trouble the leaves, but after that it was not able to continue. So that's why I just after second instar I tried the same caster. By using caster I laid. So I mentioned it here. So but first 2 instars only I mentioned the Jatropha, Indian, Badam, Arjuna, and then some other host plants I mentioned. Pixa and Acacia just to stop because it is not able to touch. All worms will be died. So like to touch. So other crops next generation continues by the. Yes, that's also interesting. Yes. So whatever crops available in forest ecosystem, I just tried because it is a polyphagous. So that's it. So, but I when compared to all crops, the tapioca is the highly alternative host plants for very silkworm ready. So we in Tamil Nadu, some areas because it is a monocrop, it is a mixed crops only we are using the castor. So it is not available. Only roadside or some other roadside only it's available. So we cannot continue for depending upon the roadside mulberry castor lakes. So just in wherever monocrop cultivable area, so Yetapur in those areas, Salem, wherever monocrop for tapioca or castor, so in those areas we can recommend the castor cultivation. So it's not it's a partial idea. For my curiosity, for interest, I did this work.
Oh, congratulations.
Thank you.
First one in you use Yatapur farm or what is
No, sir. Here locally, notes some areas they are using cultivating tapioca.
Farmers who talented. What percentage of their leaf get from them? Tapioca?
Tapioca is a whole only 100%. But preference will be compared to.
No, I am asking whether the farmer feel correct the tapioca leaf or not. Okay.
Yes.
What the percentage of leaf will utilize from the tapioca this plant?
Totally.
என்ன சொல்றாங்க the leaf எல்லாம் எடுத்து கிழங்கு வந்து அதெல்லாம் கேட்கறாங்க affecting the tuber.
Actually, it is I just discussed with them. So in some period they will just remove the leaves also. So for maturation or bigger size of root.
The sporadic study says that up to 30% we can report. Yes. Maximum can go up to 30%.
Forest colony.
Actually, after taking project with the analysis,
Yes, sir, we are.
support form and cleaning. So you told them take it.
It's student Michael Tanga. Yes, so
There is something for some project to that, yes.
In Namakkal area, Namakkal district, Namakkal Dharwar district, there are what is there? Height index not available.
You also there are.
People that really highest number of take.
But the Salem is better.
Okay, other Namakkal.
Salem, sir.
Salem.
Salem. Erode is also with the area we have, sir. And Kalakurchi, all those areas we have.
No, because we wanted you local person, na? Well, Devo is not coming forward. Kamla said to not coming forward.
Myself and Davida madam, when she came, she is the training program. That time we discussed both of us. Madam, we want to, she also told that we want to collaborate. Sir is asking for some projects of like that. Then she told that she is discussing with the persons from airport station. And we all agree together, we will make a project and we submit like that. It is in the process, but still not like that. Madam is
Getting delayed because
maybe I'll talk again, sir.
because some data the only variety, which variety then become.
Yes, sir.
It is 100 something.
That's what I suggested for screening of existing properties for feeding behavior. Sir, one thing. Second thing is what is the future management package we can go with, even if we remove the
Yes, yes.
from 30% without affecting the yield. The 2 points we discussed, we want to work on that.
We want badly, the positive conduct is
That's
quickly.
If you want, we can also give a.
No, actually the core, we report madam. Otherwise we leave the sales act difference, so under your, we can say, we have the target to come to ministry. The state. Gujarat will commit, Tamil Nadu list target.
Madam, sir, if she is with the head of station, then we can give us a trade proposal. The future management.
We make a comprehensive.
Comprehensive, is there any will do it?
They are deputy about the capital purpose.
Okay.
So that we can throw benefit.
Good, sir. Like Gujarat also now, it was not there. Gujarat already cash up front is high. About 6.5 lakh hectares they have. They are also lead post based. Now we are at least 20% income there. Now Tamil Nadu has people have the potential. I visited
Annur Satyamuniam area, people are going for Castor, and Castor is the biggest area we have in Erode district. That's an inter problem.
What Lalita? So any man who is sitting and not asking anything. Capital to done work also.
It is having high police.
Before that, I have to give them.
They are not very good ones.
Local varieties, they produce a lot of foliage and it takes a lot of time for the root formation. So within that, we can do 2 rearings with the beets. So since the root formation it is taking a lot of time, we can allow the root formation to take it to 1 year time. Immediate harvest is not get.
You can do actually. We have enough time to say that in Mysore. We stop it from their institute. That I don't stop it. Do cannot give project, we stop. From Mysore after that, the thing.
6 varieties.
No. We wanted to have local varieties of.
Local varieties.
Tamil Nadu we wanted. We want to work. We want to work. And region based work. Quickly submit, madam. By March, we submit what is that?
I will invite Dr. Sarda and his brief to talk on visualization of anti-actinobacterial organism against mulberry bleach rot pathogens.
Good morning, senior scientists, my teachers and friends. My topic is on part of no village reward. In that work, I isolated actinomyces against root rot, mulberry root rot pathogens. In coming to that exact point, here I visualized how the different microbes exhibit different mechanisms to manage and control the pathogens in the soil. In particularly, they exhibit bioactive compounds, volatile compounds, and including various mechanisms. Here we visualize how physically the actinobacteria manage to kill the pathogens in the soil in the laboratory conditions actually. First of all, I isolated more than 30 to 40 actinomyces, and in coming to that, 2 potent isolates used for this study. The potent isolates in the laboratory conditions while giving the uniform conditions, growing conditions, they interact with the pathogens. How in the sense we can see the pathogens here. The spore chains of actinomyces used to cover the pathogen spores in sclerotia as well as mycelium. And coming to that, they start to in the real world and see the invasion, hunting and behavior of wild animals or predators over the herbivore. Likewise, we also see in the microbial world. In the microbial world, the predator, or else we are calling the antifungal organisms, they exhibit the same behavior of invasion as well as hooking and coiling and squeezing like behavior of microbes to against pathogen microbes. And here we can see that clearly we can see that the pathogen structures and spores become very thin, as well as they were very fragile, and the compounds, bioactive compounds produced by the actinomyces will affect the pathogens. And finally, we see the control plates. We can see the healthy mycelium and smooth surface of the spores, as well as the sclerotia is also unaffected in the control plates. And in conclusion, we can, and once again, your mention is the antifungal mechanisms. There are multiple mechanisms. Once the potent isolate is allowed in the environment, they isolate the exhibit their mechanisms in multifarious ways. It means they produce more volatile compounds and spore structures and toxic compounds to the pathogens. In such a way, they control the pathogens in laboratory condition as well as in natural condition. Yes. Starch is
How do you detect the even in that?
Starch is segregation in fungal media, and actinomyces also the advantage in the pathogen.
And you able to grow both things.
Yes. And normally we use the fungal culture, we use starch based ingredients, but use one. So I merged it to
And how do you identify this active
random microbiology. Identify based on morphology as well as morphology, they produce
7%. What is it? Which one we have?
molecular markers PCR.
I mean, I see. Which primary is being
Yes, sir. Primary is farmers 27 sir, and the reverse is
which is the Indian.
So this I
You are going to outsourcing or
Field evaluation has to be done.
yes,
Field evaluation.
field evaluation has to be done.
Now you can recommend for that.
Don't change the emblem of the organization. Just open it.
Open.
You are in front of change.
Please open it. First it is open. Should not color it and should not-
Should not change. Color, we should not put on.
White, red line. Yes.
What is available, what is approved by government, government only will put it. You cannot change this. Okay.
So then we move on to next topic, nutritional profile of castor genotypes. That's all. That's all. Okay.
What the project